Okay, do you hear me well there, please? Oh, sorry. Okay.
Yes, we hear you well.
Once again, yeah. Okay, thank you. Okay. All right. Okay. Just to make sure here we have the okay, this is here. And I started with you last lecture with the order, I guess, called Erysipelas. What as I mentioned to you, because this order is parasitic and cause what is known, the disease known as the powdery mildews. Okay. And the example I started with you, we have two genera. This one is known as Macrospora. When you put SPP, that there are many species here, and also we have Erysipelas. Okay. I already started with you. I talked a little bit about its vegetative or somatic structure already. We talk about that. Okay. This order, as a matter of fact, is an order which is also called what you call, after doing sexual reproduction, it makes what you call clisthesia. Okay, and I think I showed you this time, which is it causes a disease known as powdery mildews on the plant, and of course this it comes over the surface. Of course, either the conidia, or if there is sexual reproduction, it becomes what you call the clisthesia. So this is known as the powdery mildews, and I think I explained to you that this fungus, as a matter of fact, when you do the asexual, usually will produce what you call the conidiophore and the conidia in a form of a chain. Okay, but of course, when you're going to do a sexual production, it's going to produce a clisthesia, which is a very well built clisthesia, and usually it has different appendages to allow it to click on the surface of the host. Okay, but as I mentioned to you, I'm going to mention again here that these appendages, as a matter of fact, they show different shapes and forms, and by this we can differentiate between the different species of this organism. Okay, just let me. Okay. Already, I think I talked with you with the somatic structure. Okay, we mentioned that about the and then I started basically with the reproduction or life cycle. Okay, and first I started with the asexual. Okay. That will I stopped with you last lecture. I'm going to continue from this point here. Of course, as I mentioned to you, basically in this one, as already you can see here, after a period of vegetative growth, okay, basically the hyphae then produce what you call a straight conidiaphore and then a chain of conidia. And of course, this conidia is very simple. Later on, it's going to be blown by the wind. And of course, when it's going to find the suitable host, is going to germinate and good. But as I mentioned to you that this conidia, as a matter of fact, for some reason or how, they can germinate even at zero relative humidity, even in very dry conditions. So basically, this is the one this looks like here, this one here. This is the one I'm going to talk about now at the moment. Okay. Are you following with this? Okay. Let's do continue like this here. All right. Now, as I mentioned to you here, basically after a period, after a period of vegetative growth, okay, as I mentioned, after a period, okay, a certain hyphae in most species certain hyphae produce erect, conidiophore, which is basically a kind of hyphae. From which form conidia in chains. That means in one chain, in chains. Okay.
Sir, could you adjust your page, please?
You see it now?
Just a little bit to the right. Yep, that's perfect.
Okay. Now, as I mentioned, basically, that they produce that conidia in chain, and of course, after that, they're blown by the wind, okay? Which blown by air current, and of course, and germinate initiating a new infection, and germinate initiating, of course, they germinate to a new hyphae, initiating, because as I mentioned to you, that this species is parasitic. That means in order to, all the time it needs the host to germinate on it, initiating, okay? Initiating what we call a new infection, okay? Just to mention here that this characteristic, that powdery mildews which is of this species. Okay, Conidia, which is basically, of course, is a little bit in contrast with a lot of other fungi, which is they need quite a bit of humidity. But this one basically, okay, Conidia, able to germinate. Okay, at 0% relative humidity. Okay. And of course, later on, the asexual cycle will repeat itself. Now we're going to move to the sexual reproduction. Sexual reproduction, basically, the steps is more or less very similar to that in which we talk about in penicillium, okay, which is very simple. Okay, as I mentioned to you here, basically what you call, and this species here, usually also the same thing. They have the gametes, which is known as ascogonium here, as you can see it here, represent the female, and the anthridium basically represent the male. And usually they are curved around each other like this, especially the anthridium. And upon the maturity, of course, what happened here, then the nuclei from the anthridium is going to move to that of the ascogonium. And of course, they're going to stay as what's called characteristic of all this group, as I mentioned to you, that stage in which we call plasmogamy, in which occur two nuclei, which is known as dikaryotic. It will stay a while until the two nuclei unite and produce a zygote. You follow me that? Because that's a characteristic of most of this group of the what you call of the dicaryomycota, okay, including of course the ascomycete. Now, of course, the ascomycete always, once the two nuclei unite, meiosis will take place, and when meiosis will take place, then it's going to produce what you call meiosis about occur 3 division. Are you following me? Occur 3 cell division. Now, as you know, meiosis, it has 2 divisions, meiosis 1 and then meiosis 2. And when you have meiosis 1 and meiosis 2, normally how many cells we get after meiosis 2? Can anybody answer me? If you have cell division, meiosis 1 and meiosis 2, at the end, how many haploid cells we're going to get? 4. Is that clear? Okay. And then when it happens, the other division, which is going to be as mitosis, is going to produce 8. And that's why each of the ascus contain 8 ascospores. Of course, during that process, it was developing, then some certain hyphae will develop, forming what you call as the clustothecium. And once the clustotecium develop, then inside the clustothecium, you may find, for example, either one ascus or more, depending on the species. Okay, and usually at the end of the season, and the ascus and the clustothecium, when it mature, basically it looks like a little bit more dark brownish in color and with appendages. So this basically presents more or less the sexual life cycle of the powdery mildews, or in this case, of course, of the erisiph, or even including the Mycospora. So now I'm going to continue with this one here. Okay. In general, I'm going to mention a few points. Usually, the sexual reproduction, okay, and ascocarp formation, or the fruiting body formation, and ascocarp. Of course, the ascocarp in this case is a kind of clustothecia, because as I mentioned, this order is a clustothecial order. Clustothecia, okay. As I mentioned, ascocarp or clustothecia formation occur later in the growing season. Okay, which is really about midsummer, which is about between two brackets. Okay. Now, the other point I'm going to mention, as in always, the six organ basically include these two short hyphae, which is known as Anthridium and Ascogonium. Okay, to mention this point here, the six organs consists, okay, of short hyphae that curve around each other, as I showed to you. That curved, okay, around each other. Okay. And of course, these identified, okay, these six colon identified, as I told you, as an Anthridium, which is simply, this is the male, represent the male, and Ascogonium. Which is basically present the female. Okay, so second here. All right, so basically, as I mentioned, usually then at the point of contact, of course, then that area of contact between the Ascogonium when they mature and the Anthridium will dissolve, and the nuclei or the nucleus migrate of the Anthridium to the cells of the Ascogonium. So just to mention this point here, at the point of contact, that means already between, I just mention here that the cell wall between the male and female hyphae, that means between the Anthridium and the Ascogonium, between the male and female hyphae, as I mentioned to you, dissolve. Okay. And of course, and the result of that, and the nucleus from Anthridium or Anthridium migrate to the Ascogonium cell. Okay. And as I mentioned to you, basically as characteristic of all this group of fungi, the two nuclei will remain for a while. Okay. So simply to mention to you here that a nuclear, okay, union, okay, is delayed. Okay. As the characteristic, don't forget that, of this group of fungi. Of course, I mean by this group of fungi that all the ascomycete and basically because within that phylum, Dicomycota. Okay? Are you following me here? Okay? Now, let's try to summarize this here. Then after this, what happened here, to mention to you, depending, just have, okay. Then the somatic hyphae around what you call the sex organ after unification, okay? Form the sterile protective layer which become okay, which become, of course, the walls of the clistothecium or clistothecium, which become the walls of the clistothecium. You follow that? Just a second here. I will explain that to you in a second here. That basically, as I mentioned to you here, if you look to this figure here, okay, as we have, this is the escogonium, and this is the anthridium after the biographic, and you have two nuclei, and of course, these nuclei go to take some time until what you call zygote develop, and then what you call meiosis occur. During a while, as I mentioned, the hyphae, which we call the somatic hyphae, or we give them sometimes a sterile hyphae because they are not involved in the what you call formation of the ascospore, which is around these ascos that they develop, and in the end that cells or this protective layer will form in the end, which is known as the clistothecium. Okay, you follow that? So basically that's what I mean here. Okay. Then, as I mentioned to you here, after unification of the male and female nucleus, okay, the zygote, well, in this case, quick, nucleus undergoes, okay, the three successive cell division or nuclear division. Okay, successive a nuclear division. Put it down here. As I mentioned to you, this nuclear division is basically meiosis. As I told you, meiosis 1 and then meiosis 2, followed by mitosis, which will end up what you call 8 spore, okay? And discuss. Okay. In each ascus, of course, resulting, okay, in 8 ascospores, a nucleus. Okay. As we are developed. Okay. Now, once you mention this one here, okay, that in each Christothecium, basically, as I mentioned, it could be one ascus or more developed, that it depends on the species. So I'm going to mention this point here, depending on the species. Okay. Formation of one or more ascus, okay, develop in each Christothecium. Okay. Now, always remember this organism, whether they develop in the asexual, I told you, the conidia, or they develop the Christothecia, always that it develop what you call on the surface of the leaf, so that of the host species, so that it can be dispersed by the wind and what you call, of course, from the organism point of view, is to reproduce itself, but in other words, it's going to what you call to increase also the infection. Okay, to mention this here. Now, as I mentioned to you here, that the Christothecia, okay, of course, I mentioned Christothecia as a plural to Christothecium, okay, usually develop on the surface of the leaf, or of the, it's better to make it of the host leaves, of the host's leaves. Okay. Now, the other point I'm going to mention to you is that usually that Christothecia, when it matures, is always dark in color. To make this point here, the Christothecium okay, just to make sure here, the Christothecium, okay, at maturity, okay, their color is darkened, okay, is darkened to brown, and really sometimes come almost like black and nearly black. Okay. The other point I'm going to mention to you here, that basically, as I mentioned to you, that the Clostridium or of the powdery mildews usually bear appendages, and usually they show different shapes. But of course, these appendages is important because that's the way they click the what you call on the surface of the leaf. I'm going to show to you. Okay, and to mention this point here, the Clostridium of mention of the powdery mildew, because that is the name of the disease they cause. Of the powdery mildews, simply to make it of the powdery mildews species, because there are different species. Okay, bear appendages. Okay. Okay. Of different types. And of course, these appendages will help what you call that organism, as a matter of fact, to cling to the surface, okay? And simply, which are useful in anchoring, which are useful in anchoring, of course, the fruiting body, or bodies here, on the leaf surfaces, or on the leaf surface. Okay. And as I mentioned to you, basically, that these two characters, the number of the asci in the Clostridium and the pattern of these appendages, it helps us to differentiate between the different species of the powdery mildew. To mention this point here, okay, that the number of asci and the Clostridium okay, and the pattern of appendages simply going to mention helps in differentiating between the species. Help in differentiating between the species. And of course, I mean between the species of the powdery mildews. So basically, this is the sexual life cycle of this, what you call, of the order, we call it either the order Erysipelas, which I give you basically the two examples, either Myrospora alani or Erysife, okay? And we discuss basically the shape of the conidia and also the, what you call, the sexual life cycle. Is that clear? Okay? After this, just give me a second here to give you this. Okay. As you can see here, basically, I think that, for example, if you have different Clostridium here, which shows you the pattern of the appendages, okay, the shape of the appendages here, the type of it, and also that some it may contain more than one ascus, other it may contain only one ascus, and of course inside it the ascus spore. And basically that is how we pull. And that shows to you, if you look to this, others even like this one here, some species look like this one here. And here basically we shows to you that a combined picture This is basically, as you can see, as a result of the asexual reproduction, you have the conidia with a chain of conidia, and this shows you here basically that the cristothecia and how it's attached to the surface of the leaf with the appendages. Okay, so basically this is with regard to this group of fungi within this ascomycete, which are important, but also it is a cristothecia type of fungi. However, one difference between this order, which I talk about the order Erysipelas, and the previous order I talk about, which is called Sorodialis, in which we have the Aspergillus and Penicillium. In Aspergillus and Penicillium, we mentioned that the cristothecia is very weakly developed, while in this one, in this order, the cristothecia is very well developed. Okay, so now going to move to another order here. Are you following this? Okay, and now going to move to another order, which is known as, this order, we're going to look about it, the new one here, is the order in which, as I mentioned, they develop their, what you call the fruiting body in a form of parathecia. You know the parathecia. The parathecia basically, as I mentioned, is that fruiting body which looks like a flask shape, okay, with an opening. Okay, so now do you finish from this one now? I'm asking you. Well, okay. Now, in this order here, I'm going to talk about is known as the order. We call it. Give me a second here. Okay. This order, one of the order I'm going to talk about here is known as the order. Okay, Sorodialis. This order known as Sorodialis. Okay, and as I told you that this order basically is a parathecial order. That means when they produce their fruiting body in a form of like flask shape like this one here, it would basically, do you see this one? In which basically you have flask shape with an opening here, which is known as the ostiole, and here you can have basically, as a matter of fact, the ascus, and between them, of course, you have the paraphyses. I'm going to show it to you again. Now, this order here, I'm just going to mention three names or three genera in this order. So basically, this is a parathecial order. Okay. Now, example, one is important to mention, is called Neurospora. Species, and the other one I'm going to mention Sorodialis species. And the third one is known as Chotimum. CH. Known as Chotimum species. Basically, I'm going to talk about these two. I'm not, and this one I'm just describe the, what you call the shape of the cristothecia. I'm not going to go more into their reproduction, okay? But also to mention to you that the importance of these, more to talk a little bit about Neurospora and Chotimum, that Neurospora, as a matter of fact, is important because as a result of the cristothecia and the size of the ascospore, they are used quite a bit, what you call, mostly in the, what you call, to developing the genetics of the fungi. And that's why basically, as you know, normally we, most of the laws of the genetic, we use what you call that fruiting fly, which is known as the Drosophila. You know that? Okay. So usually we call this as the Drosophila of the fungi because you use it that. So basically to mention this one here, a bit of general characteristic of this group. Now, as I mentioned to you that at the general here, that basically that this order, okay, include about 5 families, include 5 families, and contain about 75 genera. And 75 genera, including, of course, also about, within that, about 600 species, including, okay, 600 species. Okay. Now, the other thing I want to mention, that most members of this group, basically, they are saprophyte or saprobic. Okay. Just to mention this point, this is, meaning this group here, this is a generally saprobic group. Okay. Of fungi, and also to mention, okay, producing okay, a solitary one, solitary perithecial ascomata. So there are solitary perithecial ascomata. Okay, ascomata. And basically, as I mentioned, because as the group are saprophytic or saprobic, that means usually we are going to find them mostly in area which are decaying, like decaying plant, decaying, okay, and to mention, and found, okay, on dung and decaying plant remains, or a plant remains. Okay. Now, the other thing I want to mention in this group, basically because of their use basically as agent. Okay. Now, as I mentioned, that several members of this order okay, are important tools okay, they are important tools in fungal genetics. Okay, in fungal genetics, okay, and also and biochemistry. Okay. And basically, the most important, sorry about that. The most important one of these are basically Neurospora are used, okay? And to mention this point here, first and foremost, is basically Neurospora. Okay, is Neurospora, and basically which has been called okay as the Drosophila, as to mention here as the Drosophila, sorry. You know, Drosophila is the name of the genus of the fruiting fly or the fungus wort. Because mostly in laws of genetic we use the Drosophila, but in fungi basically this is what okay. and that we call it as a proverb, as Drosophila of the fungus worm. Before that, and now basically these are as general. I'm going to talk a little bit about it called two of these orders. One is the Neurospora, we call Neurospora stifolia, and the other one I'm going to touch on Schutima. The reason why I'm calling, because this Neurospora, basically, as a matter of fact, stifolia, oil species, is very common in bakery, okay, because they really infect a lot of bakeries and produce the dough. It looks like red, like this one here, okay? Give me a second here, okay? That basically, of course, being infected, and sometimes it's very difficult. Once being infected place, it's very difficult to get it because to the huge number of the spores, and also they are very easily multiplied. Okay. Just to mention a little bit about this fungus here. Okay, the first one known as, talk about Neurospora. Here, okay. Known as this species Neurospora stifolia. This is H here. As I mentioned to you, it's commonly known as because it affects this, and that's what they call it, the bakery mold. It's commonly known as the bakery mold. Or we call it, as I showed you, or known as, because of the color of the red, or it's known as the red, what you call, the red bread mold, or known as the red bread mold. Okay, as I mentioned, because okay, it frequently infests okay, the bakeries, and of course, once they infect, of course, going to cause quite a bit of damage because you have to, okay, and cause considerable damage. Okay. And also the same thing, sometimes basically if there is a microbiological lab or media and it's if managed to reach there and infect, it will create havoc. That means basically because of the huge number, so it's very difficult to get rid of it. It needs quite a bit of sterilization and so on. Just to mention a few lines of this one here, okay? Just to mention that when it invades, means this fungus, okay, it plays a havoc, okay, also with cultures, and as I mentioned, and it's difficult as I mentioned to you, to exterminate. Okay. As I mentioned to you, this is mainly because, okay, of the enormous or large number of the enormous quantities of easily dispersed conidia. Okay. Now just a few lines here to show the characteristic of the hyphae, only one or two lines I'm going to mention to you here. Now, its mycelium okay, it consists okay, of numerous branches okay, of hyphae. And usually when they produce their conidia, the conidia is oval, and usually as a rule, it is pinkish in color, and that's why when in fact they call that red of it. Okay. As I mentioned, hyphae easily recognized okay, by, as I mentioned to you, by the pink, as I mentioned to you, by the pink oval conidia. Oval shape, of course, conidia. Okay. Now, the other thing I'm going to mention to you with this organism, usually when they produce the conidia as a result of asexual production, the conidia basically sometimes they contain two types or two sizes, either the regular one or a little bit more, what you call, the smaller one, okay, which is known as microconidia, to mention to you. That the organism okay, produce two types of conidia, okay, the normal size okay, normal size, and of course the smaller one known as and microconidia. That means the smaller. Okay. Now, of course, when it happened, the sexual protection, I'm not going to talk about it, but just to mention, of course, they're going to produce a parathecia, and this parathecia, of course, inside what you call with the ascus and ascospore. This is to mention another point here. Okay. Very simple, going to mention, of course. Now, parathecial production as you know, this is a result of the sexual. So in that case, they require two mating type. Okay. So the parathecia of intimation requires the mating simply a mention of two strains. Okay, of two strains. Just to mention it here. Okay. Now, the other thing I want to mention, basically, when the parathecium mature, okay, just to describe it itself, okay, simply going to mention to you here that mature parathecia the mature parathecia also are dark colored. Okay. called like pyroforms. Okay. And of course, and contain the numerous octospores. Numerous octospores. Octospores. You know what means this? We call it octospores because each of these ascus, octo means 8. Okay, we call it octospores. Okay, because typically, okay, or you can call it ascospore. Okay, means ascus with 8 ascospores, but we call it octospores and numerous octospores. Okay, asci. Okay. But of course, usually the perithia itself, usually it has an opening, okay, which is already, I showed you the picture of it. That is the one here, okay. Just to, okay, second here. Okay. Now, this is basically for yourself, if you want to know, that is the perithecia. And of course, this is all of it called, known as the ascomycota or vascutina. You have the ostium, which is the opening here. And normally, of course, sometimes if we have a layer like this, in general, I'm going to give you the information here. And when you have the ascus, of course, all in one layer, it becomes like hymenium, or we call this, if it's thick, we call it stroma. Okay? And of course, here you have the ascus, and these are the ascospores, and these the sterile hyphae, as you know, between the ascus, known as paraphysis. Simple as this, okay? Now, this is basically what I talk about, the first one about the Neurospora, which is a perithecal one. The other one, the Schitumum, the reason I'm calling it, because it has, I don't know if you have seen it in the picture of the labs or so on, they have the perithecia, but the perithecia, basically, it is also what you call, with a lot of curly hairs, like this, okay, all this. And the reason we mention, because if they are occur in the air, and people breathe, of course, going to cause a lot of allergy because of this. And other characteristic of it, usually their ascospores are, as you can see, we call lemon shaped. So basically, I'm going to talk about this species, the other one here, which is of the Schitumum. Okay, just to describe it. Okay. As I mentioned, so this is what you talk about this species here. Give me a second here. Okay. Now I'm going to talk about the other example. Okay, just if you can see that properly here, you follow this. Okay. The other one is going to, the other example I'm going to use, known as Schitumum. Species. Okay. Basically, the family. Okay. Of this fungus. Of course, when you mention family, basically as a family, in that case, usually will end up with ACEA, known as Schistomaceae. Okay, just mention to you here the family, which is known as, okay, Schistomaceae. I'm going to continue here. Okay, you see that, known as Schistomaceae. Okay. And most of these members also, they are what you call as a saprobic, but they're able to use the cellulose. So in this case, you call them cellulose destroying, okay, fungi. Okay. So as I mentioned, the family of this fungus, which to include, continue here, include or includes, okay, a number, okay, of cellulose destroying fungi, of cellulose destroying fungi. And that's why basically if they infect, they can infect paper, fabrics, all that. And mentioned to you here, which grow on paper and fabrics, and that's basically if they can infect, for example, a factory or even a storage in that case, of course, going to cause quite a bit of damage. And that's what to mention this point here. Okay, and sometimes as I mentioned to you, causing considerable damage. Okay. And also the same thing, of course, as long as they take cellulose, in that case, they can grow on hay and straw and so on. And to mention to you, the straw, dung, and similar materials, are also common, as I mentioned, as a common substrate for this fungus. Simply to mention, are also common substrate for these fungi. Okay. Now, this is about this member here, just about 2 or 3 lines. I'm going to talk about what you call the one and describe what you call the perithecia itself and thing. Okay. Now, the perithecia in this group as I mentioned to you, are covered with hairs, and as I showed you, basically, in most species, they are long and curly. And just to mention to you here, which in most species, are long and curly. The other thing to mention to you, the ascus or the asci to mention, the asci, are generally, usually they are dark in color, are dark, and usually, as I mentioned to you, the ascospore are lemon shaped, and to mention to you, with lemon shaped ascospores. So the asci are dealing with, to mention to you, lemon shaped, ascospores, and of course, as the perithecia, usually they are released from that opening, which we call it the ostiole. And just to mention this point here, are released from the perithecia, through the ostiole, the ostium. So the ostium basically that opening at the end of, okay. You follow that? Okay. So basically, these are what I talk about, this order of Sorodialis, which we discussed. Basically, I gave you the name of 3 genera, the Neurospora, Sorodaria, and Schutimum. But we talk a little bit about Neurospora because it's an important, and also we talk about Schutimum. Now I'm going to move to another order, but also that order is very important, and which calls basically the ergot. Okay, but also when they produce during sexual production, where they produce the fruiting body is also in a form of paresthesia. Now this order here, known as order Calvsipterius. Now talk about this order here, Calvsipterius. Okay. And the example I'm going to mention to you here, the example known as Calvocipes purpurea. Okay. Now basically, with this fungus here, just give me a second here, okay? Just okay. Usually, this fungus is a host on, especially on rye, okay? And it is like this one here. Just give me a second. I don't know if I've seen it in the lab yet. Okay, they develop like ergot like this, okay, which is very hard brown here, and like this one looks. Okay. Now, usually, of course, this ergot of occur, of course, it's very, very contain a huge number of alkaloid and a very toxic. And especially, as I mentioned to you, during the Middle Ages, of course, they were not aware of it. Okay. And they ate basically bread was made from rye and contaminated with this. And what happened here, because that when enter the body, that alkaloid will constrict the veins. I'm going to talk about all that in details when I talk about the toxin produced by the fungi. They constrict the vein, and what happened here, of course, if you constrict the vein of in the arm or in the legs or in your hand, of course, there is no more blood, and that's why basically in the end, what you call the part of the body is going to rotten and fall off. Okay, and that's basically, it caused a lot of havoc during the Middle Ages, which are not aware. Plus, also it can cause, when people they eat this, it causes inside like a burning and hallucination, and used to call it St. Anthony fire. Going to talk about a little bit here and the effect of this and how they do the infection a little bit. So, okay, but as I mentioned, this is also Christothesia. Now, as a general, we talk about this one here and this group here. Okay, well, let's see. I have about 10 minutes or so. Time goes by. They are about, as a general, okay, there are about 27 genera, okay, and including, as I mentioned, about 270 species. Okay. And to mention basically, because the way they do this fungus is very parasitic and very highly sophisticated, because what happened here, that the conidium is going to go affect what you call inside the stigma of the flower of the rye. It goes inside and it's going to go inside the ovary and replaces the ovary. So they are very, very, okay, sophisticated in that sense of their infection. To mention to you that in this order, okay, this order comprises a group of highly evolved a group of highly evolved, as I mentioned to you, and sophisticated. Highly evolved and sophisticated, of course, they are very obligately parasitic fungi. Obligately, okay, parasitic fungi, okay. Simply to mention, of course, that this species, Calvocipes purpurea, now, as I mentioned to you, simply as a matter of fact, as I mentioned to you, the cause, okay, of ergot, called ergot, or fry, okay. Of course, rye, it is main host, but it could be another. On rye, which is I could put between two brackets, its main host. Okay, and infection. And infection, okay, takes place. Okay.
Are you able to move the paper?
Like this?
Yes, perfect. Thank you.
Sometimes I don't see it unless I focus there. Okay, so yeah, because I'm struggling. I don't like to cut me what you call. Takes place, symptomation, of course, through the stigma. I think I will stop my lecture here and I will use a few minutes just to explain to you on the figure, okay? So that also you don't rush. Okay, stigma here, and simply as a matter of fact, what happened here, that as I showed you here, just hopefully very quickly, I'm going to mention it, okay? That this is basically how the fungus will go inside the conidia and infect what you call through the stigma, of course, of the male, of the flower, and then inside the ovary, and then later on it's going to germinate and produce a lot of hyphae like this. You see this hyphae here? And with a lot of conidia. But the problem with this one, it produces a lot of sticky material, which is very sweet. And that's what happened, will attract insect, and will attract the insect, of course, they are going to propagate the fungus more and more over. Okay, and later on when that sticky material is going to harden, then it's become what you call like sclerotia, and that sclerotia, or the hardened one, we call it the ergot. Okay, so basically by this I will continue with you next lecture. Of course, this Friday is your test. I'm going to send you a little bit more detail. Of course, I'm going to send you the text through the email, through the email, and they're going to create for you a Dropbox. Okay, I show you the time, and when you finish, you can send it back to the Dropbox and I mention, okay? Or if you have a difficulty during the Zoom, because the Zoom is all the time, if you have difficulty when the Zoom is on and writing your answer, because sometimes some students, they don't have what you call, they download that program there, okay? In that case, of course, you can switch the Zoom off, okay? Okay, I will send you all this information, and if you have any question, please let me know. Are you okay? Well, it seems all right.
Yeah. Yes, I think we're pretty good.
Pretty good, that's good. Okay then, I will send you what you call the information, and also what you call the box and the time. And usually I give you at least 10 or minute extra, because sometimes the box will close. If it closes, you can send me. If you late, of course, I will deduct a little bit of mark, but at least you can send it in the end as what you call to my email. Okay, we'll talk to you and send you more information. Okay, have a good day.
Thank you.
You're welcome. when we finish early, let me do this hair a little bit.